So now we want to delve into the topic of cell signaling. And in my mind, this is really kind of an introduction to the cell as a machine. This is now the mechanism by which cells are really going to process information from their environment, and then how they get that information from the receptor to some mechanism in case of differential gene expression or the activation of an enzyme that affects the cell, that changes it. In this slide here, you just see the idea that you've got two different mating types of yeast cells, and how do they not only determine that there is the presence of an alternative strain in the local vicinity, but then move toward that particular strain. So we're going to have some receptor protein in the plasma membrane that binds to that mating factor that's released, and then what we're going to call a transduction of that information into the cell to ultimately change it, and in this case, have it grow in the direction of the cell that it's seeking. We'll go over specific examples as we move on, but in this overview, what we want to think about is introduce the idea of the interactome. So as we talk about the genome being all the genetic information in a particular cell, the interactome is a categorization, a catalog of all the interactions inside a cell, which you might imagine is tremendously large. So remember we talked about receptor-mediated endocytosis? Here is just a small number of proteins involved in that process and how they interact with each other. You'll notice in this side these hub proteins. These are proteins that interact with a lot of other proteins. They're particularly important. They're key points in this interaction. They're control points. So literally, this is now how we can process information kind of mechanistically. So again, there's nothing special, there's nothing magical about life. These are all tiny machines. And so you build these interaction networks, which can then get actually quite involved. And so this is now breaching into the field that we call bioinformatics. This was impossible before the use of computers in biology. And so to try to map out these interactions on paper was impossible. But now with a computer, you can create literally like a virtual environment where you can rotate and manipulate these maps and zoom in on the points that you want and see them in their entirety. Now, when we talk about signaling from cells, we're going to talk about local and long-distance signaling. So again, if you're a multicellular organism, oftentimes your environment is the other cells immediately adjacent to you. So in local signaling, we may be actually sending chemical signals directly from one cell to another through a cytoplasmic bridge. In the case of gap junctions in animal cells or plasmodesmata in plant cells. Or it may be two proteins interacting at the cell surface, indicating, well, hey, I'm next to you, or I'm now next to you where I wasn't before. All right. So we talk about local regulators that travel only a short distance versus hormones, the special word that we're going to use for chemical signals that travel a long distance. So you can see here, again, signals that travel through the extracellular matrix just by a short distance, we just call local signaling. So neurotransmitters, for example, and the synaptic signaling are local signaling. But long distance is where now you're dumping a chemical message into your blood vessels. And these travel throughout your body a long distance, quote unquote, to affect cells at a distance. All right. So the mechanism is ultimately going to be the same at a cellular level, but where those signals are coming from dictate whether we're local or long distance signaling. Now, again, speaking in general terms, cell signaling has three stages. And those stages consist of reception. So you have to have something that's going to detect the signal, right? So we call those oftentimes the receptor proteins on the membrane. And then that receptor protein has to ultimately send the message into the cell. That signal has to be transduced into some other part of the cell or some other mechanism where you have a response. So reception, transduction, and response are going to be our three components of a cell signal. So again, here we see the signaling molecule interacting with the receptor. That receptor then relays the information through many times a multi-step pathway in transduction. We're going to look at the details of each of these in turn. And then finally, that results in some kind of response to the cell. Now again, it can be a little tricky because sometimes that response is don't do anything. But you very deliberately told, or the cells receive a signal that says hold the course, right? Do what you're doing now. Or it may be a change in gene expression that we understand better now. It could be a change in enzymatic activity, modifying a biochemical pathway. So again, this is kind of a great summary slide, but in reality, these pathways can be quite complex. And that's important because we're going to see that the more complex they are, actually the more control you have over them. The ability to fine-tune the response is greater the more players that are involved in the process. We're going to come back and look at these again as we move forward with this topic.